there are some terminologies which we will discuss. So first is trigeminal nociceptive system. So we have been discussing about the trigeminal nerve, how the nociceptive signals are transmitted. Like the branches of the trigeminal nerve reaches the trigeminal ganglion, and from trigeminal ganglion it comes to the spinal nucleus, which I already mentioned. In that, the caudalis part receives all these nociceptive signals, and from there it goes to the thalamus, and from thalamus to the somatosensory cortex. This is trigeminal nociceptive system. And talking about spinal trigeminal nucleus caudalis, SP5C. This is the term which is used in this article, which mentions the spinal trigeminal nucleus caudalis. So when we see the spinal nucleus, this is the trigeminal nerve nuclei. We have the mesencephalic nuclei, the primary nucleus, and the lower blue one is the spinal nucleus. In that we have this coralis, intercalaris, and caudalis. So this caudalis is the term which is used in this article. This is in the brainstem. And we are just clearly mentioning about the nucleus here, oralis, the intercalaris, and the caudalis. In the caudalis also, we have three segments, the rostral caudal part is also there. So we will talk about it later. And they have mentioned about the C57BL6 wild type. While this mouse, which you see here, is a black mouse. So that letter BL describes the black mouse. It belongs to the C57, which is renowned for its ubiquity in genetic research. So this strain is characterized by their dark glossy coat, hence the moniker black 6. So this is the C57BL6. And they have talked about knockout mice. What is knockout? So when the embryo is in the blastocyst stage, they genetically modify certain DNA. The mouse embryonic stem cells genetically engineered to disrupt the gene. So they do the CRISPR-Cas9 cutting, editing, all those things are done here on a particular gene of interest. And this blastocyst is then transferred to a host female, not from the female from where the ova, the sperm was taken. It is to a different female, it is transferred to a host female, and which gives rise to a mouse which lacks that particular gene which they had blocked. So this is known as a knockout mice. And one term is used, acclimated mice. Acclimatization, we know it is getting accustomed to a particular environment. So for this animal studies, we need to keep those animals in an environment where we are going to perform the experiments. So they are mice that have been given time to adjust to a new environment before being used for experiments, and this helps reduce stress and mortality. That is the reason they are using this procedure. And superior joint space, this refers to the temperament of the joint. So when we look at the anatomy of the temperament of the joint, we have the mandibular fossa, we have the condyle here, the blue one is the articular disc. Above the articular disc is the superior synovial cavity. Below the articular disc near the condyle is the inferior synovial cavity. So this is the anatomy, and from this we can understand which is the superior joint space. And they have used one filament called von Frey filament. So this is the von Frey filament through which we can conduct electricity. So we conduct electricity. So when we want to apply just the pain conduction, we can use this. And they have used the restraining cylinder. Restraining cylinder is like this. So they keep inside a cylinder kind of thing. The experimental animals like mice are kept inside this. So when they apply the electrical signal, it withdraws. So that is a sign of testing the pain perception in animals. And there is a mouse Grimm's scale which is used in this article. So it is nothing but seeing the facial expression in animals. So how to see facial expression in animals? We have some scales here, like the orbital tightening. You can see pain is not present, pain is moderate, pain is severe. So with the orbital tightening, we can see whether they are having pain or not. Then nose bulge, that is one criteria to see. The next one is the cheek bulge. So we can see the bulging cheek and decide what kind of pain they have. Ear position as well as whisker change. So this tells about the mouse Grimm's scale, and these are the facial expressions of those animals. And they have used an instrument called dodognometer. So this is an instrument where a mouse is just allowed inside. It does chew through this and then get out. So the chewing needs biting. So if there is pain, it will take some extra time. So for that they are using this dolonometer. And they have talked about DNA methylation at the TNF gene promoter region. So a gene normally is like it has a body and it has a promoter region here. Methylation is adding the methyl groups into the promoter region. How does this methylation happen? It happens by when cytosine is converted to 5-methyl cytosine by DNA methyl transferases. When this promoter region is methylated, the transcription is inhibited. So the particular production of that gene, the expression of the gene is inhibited. When there is no methylation, which is called unmethylated, there is transcription where there is mRNA is formed and then it forms peptide and then goes to form the protein. So that is. And in statistical analysis, they have used the word confounder. What is a confounder? It is a variable that influences both the dependent and the independent variable, causing a spurious association. So for example, a study that aims to determine the effect of studying hours on exam scores may not take into account the participant's previous knowledge. So in this, the previous knowledge serves as a confounding variable, as it can impact both the dependent as well as the independent variable, like the time spent for studying and exam scores. So these are the confounding variables which is used in the statistical analysis. So now if you go into the article analysis, so we can read the title again. TNF-alpha, Tumor Necrosis Factor-alpha in Trigeminal Nociceptive system is critical for temporomandibular joint pain. What the novelty of this article was, there were previous researchers focused on TNF-alpha in other pain models but not in temporomandibular joint, and they claim that this is a novel approach in this context. And the study also employed TNF-alpha knockout mice to assess the impact of genetic deletion. So this is also an important thing which they have done in this study. And they have also studied the epigenetic mechanisms which plays the regulating role in TNF-alpha expression in the context of DMJ pain. So this is related to an extensive new area of investigation in this article. So the aim of the study was to explore the role of TNF-alpha in the trigeminal nociceptive system, particularly in relation to pain caused by inflammation in the DMJ. So they used mice that lack TNF-alpha, that's TNF-alpha knockout mice, to see if this genetic change affects pain levels. Materials and methods. So they used wild type C57B and 6 wild type and TNF-alpha knockout mice. So the two types of mice they have used, which are 8 to 10 weeks of age, and they were sourced from Jackson Laboratory. The mice were kept in standard conditions with a 12-hour light dark cycle and had access to food and water at all times. And they were acclimated in the animal facility for at least 1 week before the experiments and in the laboratory for 30 to 60 minutes prior testing. So what they did, they gave an intra-TNJ injection. They performed intra-TNJ injection of complete prone cytokine. So we talked about complete prone cytokine in the last seminar. So this is a water in oil emulsion that contains filled mycobacteria. So it causes inflammation. They injected this CFA into the superior joint space of the TNJ bilaterally. So it was injected bilaterally. And they did the behavioral testing. So several tests were conducted to assess pain and sensitivity. In that, they tested the orofacial mechanical hypersensitivity. Like they used calibrated one-way filaments to measure the sensitivity of the skin areas innervated by the trigeminal nerve. The mice were placed in the restraint cylinder and filaments were applied on their skin to determine the force at which they withdrew their heads. And they did the mouse Grenade scale, and this method involved recording videos of the mice. So that is, we cannot sit and watch with them. They record the videos and then assess the facial expression. So specific facial features were scored to quantify the pain level. Then they tested the functional allodynia test, like by using the dolonometer, which is used to measure the time it took for the mice to gnaw through the two doubles. There are two doubles assessing their oral function and pain response. So this is behavioral testing. And next they did Western blotting. After sacrificing the mice at various time points of injection, post-injection, the researchers harvested tissues from trigeminal ganglia and the SP5C, which I already mentioned. So these are the two tissues which were taken for analysis, and analyze expression of TNF-alpha and its receptor using quantitative Western blotting with beta-actin as a loading control. Beta-actin control. And they did a immunohistochemistry which involved double immunofluorescence staining to visualize the co-expression of TNF-alpha with specific cell markers in the two tissues which they had taken, and this helped identify the types of cells involved in the inflammatory response. And they also found the methylated DNA immunoprecipitation assay. So this assay was used to analyze the DNA methylation at the TNF gene promoter region, providing insights into the epigenetic regulation of TNF-alpha expression. Epigenetic is that methylation, acetylation, all those things are epigenetic regulation in the DNA. And the statistical analysis was performed with a mean and standard error of mean, and one-way analysis was performed for Western blotting data, and two-way ANOVA with repeated measures was performed for behavioral testing. One-way ANOVA we used to compare before and after the same individual. Two-way ANOVA we used when multiple variables will be measuring. And the student Newman-Kuhn's method was used for post-hoc test of ANOVA. When there is a significant result in the comparison of means, they do the post-hoc test. The unpaired student test was used to analyze data from the immunostaining results, and the results were considered statistically significant when the p is less than 0.05. So coming to the results, the CFA induces inflammatory pain. So the study found that injecting complete CFA into the temporomandibular joint of wild-type mice led to significant pain. So here we see the results of the wild-type mice where they injected saline and the CFA. So these are the results which they found. And there was increased TNF-alpha expression after CFA injection. The levels of TNF-alpha were found to be significantly higher in both the tissues they have taken, and the increase was observed at different time points, particularly notable at 2 hours and 1 day, and on day 4 post-injection. So this is the study result. So they compared beta-actin as control for both. This is for trigeminal ganglion and this is for the nucleus cordatus. And they have this statistical plot. And the impact of TNF-alpha knockout. So mice that lack TNF-alpha showed a marked reduction in pain. So which shows TNF-alpha plays an important role in pain. So for that they had used the knockout mice, and they did all this behavioral analysis, and they have given the analysis, like we see all the 3 branches and the orbital tightening, nose bulge, all those parameters they have measured. And they also saw the co-expression of TNF-alpha, what all are co-expressed. They had a LBA1, which is a marker for microglia and macrophages, and they had also seen MUN, which is a marker for neurons, which is co-expressed in this. They had a control group and the CFA group, and they found the co-expression. And there was DNA methylation change. So they found that CFA injection reduced the DNA methylation. So when there is reduction in DNA methylation, which means transcription happens and TNF-alpha is produced and causes pain. So that also they studied. And CFA diminished DNA methylation on day 4 post-injection in the TG, specifically at the TNF gene promoter. This is what they found. So in conclusion, the study concluded that tumor necrosis factor-alpha plays a crucial role in the development of inflammatory pain associated with temporomandibular joint disorders, and it is essential for TNF-alpha for pain mechanisms activated by CFA injection, highlighting its potential as a target for pain management. And TNF-alpha may have a complex role in modulating pain through interactions with various cell types in the nervous system, as we mentioned, the neuro-N and the microglial cells. DNA methylation could be a pathway through which inflammation affects pain perception. And targeting TNF-alpha or its regulatory pathways could lead to therapeutic strategies for treating inflammatory pain in TME disorders, and drugs that inhibit TNF-alpha or modify its expression through epigenetic mechanisms. So there were some limitations in this study because they had focused only on animal models. It was a limited sample size and short observation period, lack of behavioral diversity and absence of human data. And there were potential confounding factors like effects of stress, diet or environmental conditions on pain responses in the animal models was there. Future scope was investigating long-term effects, examine human relevance and explore other pain mechanisms, targeting epigenetic regulation and investigating the role of other receptors, and assess behavioral and emotional aspects as well. Here the emotional aspect they did not go deep into. So all these things, is the future scope of this study.